I'm Kumar. I'm currently working as a assistant professor, and I'm also leading the bioinformatics core at Science Institute and Temple University. So today I'm going to present our lab research work where we checked how DNA methylation changes occur between cancer and age-associated disorders. So before I go through deep, some of the hallmarks that are known for aging includes genomic instability and telomeres and epigenetic alterations and lots of proteoses and deregulated nutrients and mitochondrial dysfunctions and cellular senescence and the cell exhaustion and altered intercellular communication. So these are some of the major hallmarks known for aging. Apart from the hallmarks known for aging, there are some of the hallmarks which are known for cancer too, which includes self-sufficient growth signals, apoptosis, and anti-growth signals, and angiogenesis, and metastasis, and replicative potential. And recent studies showed that there is a majority of these hallmarks share with each other. Some may be divergent and some may be common. And in our lab, we mainly focus on epigenetic alterations, which show an overlap between aging and cancer. Specifically, among all the epigenetic alterations, we mainly focus on DNA methylation, where we see the global methylation against the aging, and we see whether this methylation is also same like hyper or hypomethylated among the cancer-specific genes also. So recently, according to the NCI, National Cancer Institute SEED study, age is one of the major factor which plays a key role among all the races and all the sexes, and for all the cancers. You can see here for age group between 35 to 84, there is a huge decline, huge growth of cancers, and which is directly says that yes, age plays an important role among the cancers. That's why we pick age as one of the key factors for our study. So among all the cancers, we have selected as breast cancer as one of our study for interest, because according to cancer statistics this year, this is one of the major the topmost cancer, which is almost like 30% of the women around the world are exposed to. And there are several common risk factors for breast cancer. Some of these includes familial and hormonal, genetic and environmental factors. And the recent studies, these are the known risk factors for years for all the for breast cancer work. But recently, aging also was added as one of the risk factor in breast cancer, and which lead us to take our research interest to see how the correlation between aging and breast cancer. So further, studies showed that among all races, breast cancer also shows race-specific disparity between African Americans and whites. You can see here the survival rate for the African American African American people is very low for all subtypes of breast cancer compared to the whites, indicating that this is one of the important disparity cancer between African Americans and whites. Apart from this, you can see here hazard ratio also differs with the age. So with this, we came into the conclusion that yes, breast cancer has race-specific disparities, and age also plays an important role in it. So there are several age-associated disorders and comorbidities are known. One of the major comorbidities which is known to show along with breast cancer, or which overlaps major of the risk factors, is cardiovascular disease, where characteristics such as factors such as age, diet, familial history, alcohol and obesity, hormonal replacement, tobacco, and physical activity overlap with breast cancer. Apart from cardiovascular, there is another age-associated disorder, which is called as metabolic syndrome, which has a different signaling pathways that may lead to prone to breast cancer. So for our study, we have selected metabolic syndrome for our age-associated disorder of our interest. So what is this metabolic syndrome is that it's a clinical condition where a person who has a metabolic syndrome has elevated fasting blood glucose, obesity, elevated serum fasting triglycerides, and low high density lipoprotein cholesterol, and elevated blood pressure. Why metabolic syndrome is such a significant disease is that it is also not only it is known to be risk for breast cancer is also known for risk for cardiovascular disease under other breast cancer, other cancer types, and it is also plays important role in premature mortality. And further, it is one of the most age-associated disorder, which is almost covering 34% of the US adults are infected with that. And among these, majority of them are African women with almost 38.8%. And these African women who are exposed with are from lower education, and it is one of the important driver of health disparities. That's why we selected metabolic syndrome as our age-associated disorder of our interest to see how much CpG sites it's overlap with the breast cancer. So some of the common factors that are known for causing the metabolic syndrome are genetic, environmental and social determinants, and epigenetic factors. Still, how these genomic factors that drive this whole entity, like elevated fasting blood glucose or obesity or triglyceride HDL, is not known yet. And how these genomic factors are influenced by covariates such as age, sex, race, and poverty status. So we put forth the question saying that how this metabolic syndrome is influenced by these covariates along with epigenetics. Among all the different mechanisms for epigenetic modifications for leading a person from low metabolic syndrome susceptibility to high metabolic syndrome susceptibility, methylation such as DNA methylation plays an important role. So that's why we pick DNA methylation for our study. So DNA methylation is nothing but a epigenetic modification where a methyl group will be added to the DNA, which leads to activity or suppression of the genes next to that. So that's why we are more interested to see how epigenetic modifications in metabolic syndrome. Further, so there are different definitions or scales has been defined for metabolic syndrome. One of the more major scales that are defined is by WHO, EGIR, and NCEP and ATP3, ASE, and IDF. Among all these, we have selected NCEP ATP3 classification. According to this NCEP classification, if a person has any of these three amount of parameters, he is considered as metabolic syndrome positive. So in this way, what we did, we classified our participants into metabolic syndrome yes, if they satisfy three of these following criteria or more. And if any of them doesn't satisfy, for example, a person has only one criteria or two criteria, then we consider them as no. Suppose if any of the participant doesn't have any of these character, if he has any missing criteria, we completely removed him from our analysis. So majority of our study we have conducted are the participants from our cohort, which is named as Handel's cohort. So Handel's cohort is it stands for Healthy Aging in Neighborhood Diversity Across the Lifespan, which is started by our group in 2004. And this Handel's cohort, we have 5 waves where we collect the samples from the participants on daily basis. And right now we are going under wave 5, which is between 2017 to 2021. So whatever the samples we have selected for this study are from wave 1, that is between August 2004 to March 2009. So among these selected participants, the average age of the participants we selected are between 30 to 64 years of age. And as I said, our Handel's cohort is a big cohort. Like we have like 3,720 participants, where we have 1,522 among them with the white people, and among them 2,198 are from African American ancestry. So among these samples, we have selected 458 samples, which meet our NCEP ATP3 classification, which I showed in the previous slide. And in these 458 samples, we have 142 samples, which were 142 participants who are metabolic syndrome yes, and we had 316 participants which are metabolic syndrome negative no. So among these, we have 225 participants who are from African Americans and 233 participants from whites. So further, you can see in this, these are some of the, like we have like 50 participants who are metabolic syndrome yes among African Americans, and 175 who doesn't have metabolic syndrome among these 225. And we have 92 who has metabolic syndrome positive, and who has metabolic syndrome negative, almost 141. So with this, further we have performed demographics to see what is the, as I said before, our covariates of interest were race, age, sex, and poverty status. So we did a chi-square test to see which of these variables are most interest. You can see here between the African Americans and whites or race, the p-value is less than 0.001, which is more significant, and the age is also more significant. So considering this as a factor, we stratified our samples into African Americans and whites separately, and this is the pipeline we have done after that. So after we stratified them, what we did, we collected as I said, we are collecting the DNA from regular basis. From the DNA samples, what we did, we have isolated the DNA and performed EPIC chip for methylation. So this EPIC chip profiles almost like 800,000 CpG sites at a time. So once we finish done, we will get an IDAT files. Once we get that, then we start analyzing the data. So before that, first we have pre-processed that data for quality control using WinFi package. Then we perform normalization using Noob. Then what we did, we removed the single nucleotide polymorphisms and batch effects using Combat, because we don't want to have any genetic effects on that. That's why we removed the single nucleotide polymorphisms. Then once we done that, then we performed differential methylation positions and differential methylated region analysis using regression models. So this is the results we got. If you can see, it is the volcano plot where the black color dots represents the DMPs which are not significant, and the blue color regions are the ones which has a p-value which are significant at a p-value less than 0.01, and the green color ones are the ones which are hypermethylated, and red ones are the ones which are hypomethylated. For participants where we have selected regression between metabolic syndrome and versus non-metabolic syndrome, this is the results we got for African Americans, and this is the ones DMPs we got for whites. We wanted to see how much of these DMPs overlap between them. For that, we performed Venn analysis. You can see here there was a 15% overlap between those African Americans and whites among the DMPs, but there is almost 76% of them were unique, which are specific for whites, and 9% were specific for African Americans, which indicates that there is a race-specific methylation difference between African Americans and whites when we consider metabolic syndrome as a factor. Further, we wanted to see how many of them are hypo and hypermethylated. You can see here in this, for hypermethylated, we have like 10.5% overlap, and for hypomethylated, we have like 8% overlap. Among these, the more uniqueness was present in hypermethylated for whites compared to the hypomethylated. And in case of hyper, African Americans has a low uniqueness compared to hypomethylated. With this, we came into the conclusion that hypermethylation plays a more role in whites compared to the hypomethylation, but is a reverse in case of African Americans. Further, as I said, we have selected age, race, and sex as our cofactors. So next, we considered age as our covariate of interest, and then did the same analysis and to see the difference between African Americans and whites. You can see here, the majority of the DMPs are overlapped between African Americans and white, and it was almost 58.7%, which is comparatively more compared to DMPs, which I showed for metabolic syndrome. And the uniqueness percentage is also low, indicating that age plays a major role in case of African Americans compared to the whites, which overlaps with our previous results I showed in the beginning introduction. So further, we wanted to see how many of these are hyper or hypomethylated. You can see here in hypermethylated, we have 10% uniqueness for African Americans, and only 41.4% uniqueness for whites, and there is 48.3 overlap. Further, for hypo, there is just 8.9% overlap for the whites. Excuse me, and there is a 42.4% overlap for African Americans, indicating that hypomethylation plays a major role for defining the age-specific DNA methylation for African Americans in case of metabolic syndrome. So then we proceed our next step to see how it influences the poverty status. So we compared metabolic syndrome participants who has variables poverty status above and below. And here, for poverty status, we define here based on our demographic Baltimore information, which we showed in our previous publication. So based on that, We classified our metabolic syndrome participants into poverty status above and below, and perform the same DMP analysis. You can see here, in case of poverty status, there is higher percentage of uniqueness for whites compared to the African Americans. So with this, we conclude that the majority of the whites who are exposed to metabolic syndrome are exposed to more poverty compared to the African Americans. And among these, you can see here, majority of them are hypermethylated than hypomethylated. So with this, we conclude that in metabolic syndrome, for whites, poverty status plays a key role compared to the African Americans. Further, as everybody knows, sex also plays a key role in methylation. So we checked with the sex too, where we compared methylation difference between male versus the female. And as everybody knows that sex is one of the key factors. So obviously there will be a major overlap between the African Americans and whites. But our strange, actually, African Americans has a lower percentage of methylation compared to the whites. And specifically, they have lower percentage of hypermethylation compared to the hypomethylation. So these are the DMPs analysis we have done for the covariates. From this, we conclude that yes, age, race, and sex plays a key role for methylation in case of metabolic syndrome. Further, we wanted to see whether this methylation plays a key role at the cell composition level too. Then what we did, we took the DNA samples and classified them into 5 different blood cell types. One is CD4T, and another is CD8T, another is NK cells and B cells and monocytes. And we wanted to see how this cell, at the cell composition level, how the methylation differs between African Americans and whites. You can see here the correlation values between these cells between African Americans and whites. It is not similar, it is completely different, indicating that yes, there is race-specific disparities between African Americans and whites at the cell composition level also. Further, we wanted to see how this methylation impacts at the epigenome-wide level. So for that, what we did, we performed epigenome-wide association study, where each CpG site is in each cell is compared to and correlated with against total number of cells and the total number of CpG sites. To that end, these are the results we got. In this one, the genes which are falling above the blue line are the ones which are most significant ones. And you can see here in African Americans, we have at least 6 genes which are more significant. But in case of whites, we didn't got many, and we didn't got any at all, indicating that the epigenome-wide association is more significant at the cell composition level for African Americans. Then we wanted to see, how this epigenome-wide association is influenced by age, poverty status, and sex together. And for that, what we did actually, we took all the CpG sites in African Americans and whites separately, and we added those CpGs versus these covariates together, and checked how they influenced by the regressions. So you can see here, in this case, there are some of the genes which are significant for African Americans, and there are many genes significant for whites too. For our special interest, we got this gene which is most significant at a highest threshold, about almost like 5e⁴, and this gene is a TXN1P, which is known to play a role in diabetes. So with this, we came into the conclusion now, like metabolic syndrome also have comorbidity with diabetes too. So now, right now, we are validating this in our lab using PCR to see how this expression is there, and we are also planning to extend this experiments to RNA-seq level too. So these are the results we got for epigenome-wide association. Then we are interested to see how the functional difference between the African Americans and whites exists. So for that, what we did, we performed a gene ontology analysis, where you can see the thick colored blue regions represents the molecular function, and the light blue represents the cellular components, and violet color represents the biological process. You can see here for metabolic syndrome, the top ones are for molecular functions, but you can see here for whites it is completely different. Same as here for African Americans, the cellular component for metabolic syndrome is completely, some of them are overlapping, some of them are completely different from that. Same, we saw the same for the biological process also. You can see here there is no nitrogen compound metabolic process here, but we can see here compound metabolic process here. So with this, we conclude that at functional level also there is a huge difference between African Americans and whites. Further, we did the same for the age also. For the age also, we found the same difference in between these three categories, like molecular function and cell components, and further the biological process. And to our interest, some of the major thing we observed is that in African Americans we didn't find metabolic process as one of the key cellular biological process, but we found that in whites. So you can see here metabolic process was one of the significant functional side for whites, which is missing here. Then we did the same for other covariates also, like poverty status and sex too. So you can see in case of poverty status, the top mechanism is calcium binding, which is not there for whites. And at the cellular component level, you can see here there is no intrinsic or intrinsic factors here, which is there here. Cellular component is there, and cell level function is there here, but which is missing here. The same as here also. We see here there is no nervous system development, but it is present here, indicating that for poverty status also there is a huge difference in functionality between African Americans and whites. Further, as I said, we checked for sex too, and in this overall we found one interesting thing, that the biological. So we didn't found much difference. The only difference we found is that the system development, which is there in African Americans, which is missing in case of whites. From this, overall, we conclude that for age, sex, and poverty status, there is a huge difference in functionality or biological pathways at the molecular function and cellular component biological process level between African Americans and whites. Further, the whole essence of our project is to see how many of the DNA methylation sites or CpG sites overlap between the metabolic syndrome and the breast cancer. So for that, what we did is that we downloaded the breast cancer data sets from TCGA. So in TCGA, we have different sort of data sets from different races. Like you can see here, the light green represents the methylation data sets from Asians, and green represents the data sets from blacks or African American participants, and thick blue represents these are non-reported, means they don't have a proper race information for that, and purple represents the ones from whites. Since whole of our work is to see the difference between African Americans and whites, so we have selected only these two samples. And in these samples, we selected 160 participants from African Americans, and we selected 578 participants from whites. And then we performed a DMR analysis, and then we saw how much of these overlap with our metabolic syndrome. You can see here almost like 0.54%, like 576 CpG sites overlapped between metabolic syndrome and breast cancer. So these CpGs, what we consider as a driver CpGs, which play a key role showing the pathogenesis from metabolic syndrome to breast cancer. Right now, in our lab, we are validating this, and we are developing a deep learning and machine learning algorithm to see how these CpG sites really influences the comorbidity status between metabolic syndrome and breast cancer. So among these overlapping 0.4%, there is 0.09% of them were hypermethylated with overlap, and there is 0.008% were hypomethylated. Further, in case of whites, we got like a 4.1%. You can see here more overlap in whites compared to the African Americans. Among these total overlapped ones, there is only among these overlap 3.1% were from hypermethylated, and total 0.2% were hypomethylated. So indicating that majority of the overlap between CpGs between metabolic syndrome and breast cancer are hypermethylated, no matter whether it is African Americans or whites. You can see here for African Americans it is 0.09, which is more than hypomethylated, and you can see here for whites also it is 3.1%, which is more than the 0.2% in case of hypomethylated. So overall, with this we conclude that hypermethylation of some of the CpGs may lead to pathogenesis of from metabolic syndrome to breast cancer. So overall, our study summarizes that yes, metabolic syndrome plays a distinction pattern of differential methylation between African Americans and whites, and this distinctive pattern of methylation is also different at cell type level too. And these are also there is a major difference in functional pathways too. And some of these differentially methylated patterns or CpGs plays a key role in understanding our early diagnosis and comorbidity status between metabolic syndrome and breast cancer. So these are some of the papers we published using this data. And if you find this information is interesting, you can go through these papers. And we have other papers also we have published because of time slot. I didn't explain that. So these are the majority of the data was collected from our handles cohort at NIH, National Institute of Aging. So these are the some of our people who work really hard to collect the samples for us and bring us data using the handles. So I would like to thank our Dr. Michelle Evans, who is the Deputy Scientific Director at NIA, who helped me in this collaboration. And this is our Fels Institute Bioinformatics core, where we have supercomputing facility, where I'm in charge of that. So these are our lab members, who some of them who has contributed. And this is our one of the collaborator and my PhD mentor, Dr. Srinita at SVNST. And these are some of the grants right now I have. So this whole project is funded by U54 NCI collaborate. NCI is a pilot grant for which I'm one of the PI. So, and this is also funded by RO3 and the Research Computing Grant. And I would like to thank NIA and NIH for their grant, for their support by providing me fund and all those support. And I would like to thank Sciinov for providing me this opportunity to present my